give it to someone else. How complex is it?
How complex is brain circuitry? Larry.
Thanks. Well, my talk is going to be the exact opposite end of the spectrum of Terry's. I mean, I'm up today up at the top of that scale of complexity, talking about the human brain, basically. I don't think I've run into anybody yet that doesn't agree it's the most complex, substance in the universe, pound for pound. And so what I've been thinking a lot about actually is how to deal with this complexity. And I think these numbers are completely familiar to you, but and they're very rough estimates. But you know that the human brain probably has about 10 to the 11th neurons, maybe 10 to the 14th synapses, and people have estimated in the rat brain there's about 3 orders of magnitude less each, and the human brain weighs about 1,500 grams per rat. Well, obviously this is way beyond that. We need to figure out some organizing principles, simplifying principles, etc. So one of the really basic questions to get at is what is the basic wiring diagram of the brain, the nervous system. And what that thought of, let's say, is a biological computer. And I think with that, one way to approach this is to ask what are the basic parts of the brain and how are they interconnected. And that's a classic time-honored way to think about how the machines work. And what I want to do today actually is stress the importance of an accurate physical model of the brain to start with. And here we are on the 50th anniversary of Watson and Crick. And they actually figure out the structure of DNA, basically got tinker toys and put together physically a model that they could look at and feel. And when they got to this point in 1953, the basic logic of DNA was clear. And it took 50 years, and now 50 years later, all 3 billion sequences have basically been worked out. We have a basic map of human chromosomes. But it was fundamentally based on understanding the logic of the structure. Well, it would be great to have the same thing for the brain, some basic physical model of the basic wiring diagram of the brain. There is no such thing. When you go and study neuroscience and you start reading Candell and Schwartz or however you start, there is never a discussion of the basic wiring diagram of the brain because nobody's got it. But one of the really important things about Watson and Crick and the sequencing of the human genome is that it gave boundary conditions for the basic problem of the complexity of the genome. And we now can say there are on the order of 30,000 genes. So it would be nice to, so what I've been thinking about is the basic boundary conditions of neuronal complexity, the complexity of the wiring diagram of the human brain. So I want to go through a little bit of the logic here. These are very, very rough estimates. Hopefully, order of magnitude estimates are more or less correct, but it's been very, very informative to have the basic boundary conditions on the human genome. And as I say, starting with this beautiful structural model of DNA 50 years ago. Well, as a matter of fact, the structural organization of the body was basically, most of the body was worked out 450 years ago by the sailors. And we have, analogous to the model of DNA, we have a very detailed, good model of things like the skeletal motor system. We have a great model of the circuitry system that's hardly worked out. Pavlov worked out the digestive system. We just don't have an understanding at the systems level like that for the human brain, or the brain in general, why is that? And how to get at that. Well, so what I'd like to talk about today a little bit is some of these boundary conditions and actually estimate, how many macro connections are there in the brain, in the mammalian brain. And what I mean by a connection in this context is a macro connection. It is a pathway between 2 cell groups. It's like retina to lateral connective. It's like lateral connective to area 17. These are macro connections. In contrast to the micro connections of individual neurons, where you actually have, let's say, the actual number of synapses. So to a first order approximation, the macro circuitry is determined genetically, and the micro circuitry is partly determined by experience. Actually, so what I'm talking about really is the big picture macro circuitry. And what you really want to know is how many basic parts are there, how many cell groups, recognizable cell groups, how many cell types are there, and then finally, how many connections are there per cell type. And if you think about it, that will actually give you the basic complexity of the circuit. And when I say cell group or cell part, cell type, let me just say exactly what I'm talking about. Something like this. This is a photograph of Brodmann's in 1909. It shows the difference between area 17 on the left and 18 on the right. I mean, those are very recognizable cell groups or areas. And people have gone through and you wonder how many of those are there. You know, here's another beautiful example, actually, from Hall's work a few years earlier, just showing the structural difference between the human auditory and visual cortex. Those are areas. Those are two different areas. Now, in terms of cell types, obviously cell types make up every cell group, and every cell group or area that I know of has more than one cell type in it. And what I mean specifically by cell types are the kinds of things that everybody knows about in the retina or cerebellum, where the retina has 5 basic cell types. There are a lot of varieties, but there are 5 basic cell types, and the cerebellum has 6 basic cell types and so on. Those are the best known. It gets worse from there. That's what I mean by cell type. A photoreceptor, a bipolar cell, a ganglion cell, horizontal amplitude cells. They're really defined on their location, distribution, and the distribution of their axon. What they do, where they go. Okay, so here comes the rough calculation. There are, and this is based on a lot of experience over about, well, now it's 150 years of research with these methods on a lot of different mammals. In the mammal, there are about 500,000 regions or parts, cell areas, like they're on the order of 50 cortical areas, there are cranial nerve nuclei, so on and so forth. So the best estimate there is there's about 500 to 1,000 regions of the brain that are defined by a specific set of cell types. And if we just take the best known regions, like the retina and the cerebellum, that each have 5 or 6 cell types, let's just say, for the sake of argument, that each one of those regions has about 5 cell types. What that gives you is on the order of 2,500 to 5,000 clearly different neuronal cell types. Well, the next question is the amygdala has 20 different cell groups in it. Hypothalamus has about 100. The thalamus has about 50. You know, that's how they get that.
More areas of the hypothalamus than the cortex?
Many more. You know, without getting into subdivisions and so on. Then you get into, well, if you take a neuronal cell type, how many different cell groups does this axon go to? And I don't think there's a single cell type that only goes to one. There's always collateralization that go to multiple places. And again, this is a very rough estimate. Just based on my own experience, I'd say it would be reasonable to estimate there are 10 to 20 targets, 10 to 20 different cell groups that each cell type innervates. There are examples of 2, and there are examples for several hundred. Like the locus coeruleus comes with many, many things. The supraoptic nucleus, the hypothalamus goes to. So there's a huge range, but on the average, let's just say 10 to 20. What that gives you is an estimate of 25 to 100,000 macro connections of the mammalian brain. That's what the basic circuitry consists of. Interesting, there's about 30,000 genes. It's the same order of magnitude of complexity. Now, you often hear that everything is connected to everything else in the brain. Well, if that were true, there would be 5 million connections. Let's say if there were 5,000 cell types, there would be 25 million possible connections in those cell groups. Well, as a matter of fact, there's only 100,000. So as a matter of fact, the primary connectivity of the brain is actually pretty sparse. There's a lot of divergence as you get out, but fundamentally, when you get down to A goes to B type things, this is my estimate of what we're dealing with. Now, having said that, that's all very theoretical. When you get down to the data now, how many of these do we know right now, for example, is a good question. where do we stand in terms of figuring this out? Do we have enough data to put together a model? What is the data? And that's something I've been doing for a long time. I'm basically a neuroanatomist. And here's a shocking thing. And I went through the parts of the brain that I really know, like the hypothalamus and the hippocampus. And you can take actually any of those. But let me just start with the hypothalamus as an example. And how many connections are there to the hypothalamus that we know about? This is where you get into what I'm going to be leading into next is we need a database and a knowledge management system for all of this. And one of the main reasons is, if you look at, as I say, there's a long history of this. In 1940, there were two beautiful reviews of the literature based on experiment on neuroanatomy. And they concluded that there's a total of about 55 macro connections known at that point, established for hypothalamic cell groups, inputs and outputs. Well, by today's criteria, 82% of those were false positives. So actually, they were method artifacts. They weren't even guessing right. It wasn't even chance. Almost everything they said was wrong. And that's because of the complexity of the brain. They were looking in there at these very unpowerful methods and just making stuff up. So actually almost everything was wrong. They couldn't really tell where things started and ended. Now this is circular, what we're going is by today's standards, obviously. But then now they came along with this wonderful method in 1953, and people re-looked at the whole, it's a degeneration, re-looked at the whole nervous system, basically. So for the hypothalamus, there were a couple of great reviews around 1970 when I went to grad school. Well, I went up to about 75 connections, and by today's criteria, actually half of them were reliable and valid, which means the neuroanatomy I learned in grad school is wrong, is misleading, But the trouble is a lot of it, part of the trouble is a lot of this stuff is still in literature. How do you weed out what's right on? Well, then there was this incredible revolution in neuroanatomy in about 1970 where people took advantage of cell biology, where they had used axonal transport, immunohistochemistry, and C2 hybridization. They did all of this stuff, and this is just exactly when I got into this. I reviewed the literature on the hypothalamus in 1987, and it went up to about 450 connections, and roughly 90% of those are probably valid. Then, even better techniques came along, PHAL, dye, and so on. When I tried to estimate this about 2 years ago, it was up to about 2,000 connections to the hypothalamus, and probably 95%, accurate and reliable. And when you add up the limbic system, the part of the brain that controls the emotions and drives and motivation and so on, you're up to about 4,000 connections that have been published and established so far. Almost all of them in the last 15 years. Well, going through textbooks and so on for the last 100 years, it turns out that these connections in the textbooks and everything, in almost all cases, about 15% of the total connections they find. So, if this is, say, 15% of the total, that's, 28,000 or 30,000 or so. So, maybe we know half of all of them. Now, if it's at 25,000, we're actually getting, in this particular system, maybe 3/4 of the way there to knowing, actually, not knowing most of the connections at a very crude level. And I must say, that does not apply to most of the rest of the brain. And it certainly doesn't apply to the human brain, where there is no data, and there probably never will be. So, everything that you think about the human brain has to come from animal research, whether it's right or wrong. But so anyway, this actually is where we're at right now. And I actually don't want to overload you with a lot of stuff. I'll leave some time for questions. But if you want to read more about this, Mihai Bota, Hanwei Zhang, and I published a paper in Neuroscience on this last year, where we described the knowledge management system for brain architecture, basically for connections and so on. And so you can visit that site. Mihai is still in the lab. Hanwei is now the senior neuroanatomist at the Allen Brain Institute in Seattle. And Gully Burns is also in the lab. He got started actually part of the Michael Arden's Human Brain Project. He's got an RO1, and he's got another knowledge management system. So these are two knowledge management systems that are certainly not due to me, but maybe being around has helped form them a little bit. And then actually, if you're interested in a feeble attempt at a basic wiring diagram of the nervous system, I have a book looking at biological brain architecture, where I actually try to put those 25,000 connections into 4 bins and say there are 4 basic parts that are interconnected in such and such. It's a stab at making a crude attempt at simplifying this thing, actually just about as far as you ever could. So actually you could take all 500 parts of the brain and put them into one of those 4 categories, and the functional systems fall out. Anyhow, I think I'll leave it at that. Now, the reason I present this, actually, more than anything else, is to get feedback, because, every time this is just a complete work in progress. I'm thinking about it all the time, and every time I talk about it, people come up with really good questions and, comments and ideas. Thanks.